Wonderful. So yeah, thank you so much for inviting me to speak today. I'm super excited to tell you about really the scientific framework that is the basis for the work that I'll be doing using the SETBP1 disorder in mice. And so the talk will basically be, I'll tell you about that, the basis for the reason why that I'm studying SETBP1 and in particular the circuit that I will tell you about. And then I'll tell you about the studies that I have planned. So, neurodevelopmental disorders are common, but the individual etiologies or the individual causes for those neurodevelopmental disorders are in themselves rare. Like Haley just said, we're talking about less than 200 people for SETBP1 disorder known today. And so one challenge that we have for developing therapies for neurodevelopmental disorders is that each of those genetic changes is really unique and affects the way that neurons function in different ways. And so we could potentially create therapies for each one of those genetic disorders. But I feel that that approach is going to benefit people with those specific genetic disorders. My sort of vision is that we will be able to develop therapies for neurodevelopmental disorders regardless of that underlying etiology. So one of the big sort of bottlenecks here is how do you get from these multiple different genetic causes that affect things like DNA synthesis and ion channel function and cell adhesion. How do you get from all of those varied genetic etiologies to these common behavioral symptoms that we see in our kids with neurodevelopmental disorders? So challenges with social interactions, challenges with attention. Well, as a neurologist, the approach that I take is to localize. We talk about localizing the lesion in neurology. And what that means is we try to find the place in the nervous system where the problem is. And then once we know where the problem is, then we can talk about a treatment. And so currently our ability to really localize these behavioral symptoms has really been hampered by the fact that they really live in distributed brain networks. There's not sort of one spot in the brain that we can point to that's responsible for any of these neuropsychiatric symptoms. And so in order to localize effectively to these more distributed systems, we have had to wait for technology to sort of provide us the tools to be able to do that. And about 10 years ago, we now have that tool, and it's called Optogenetics. So I'll tell you a little bit about that for those of you who aren't neuroscientists. But basically, it's allowing us to say, okay, we have these multiple different genetic changes that lead to a common behavior. What is the circuit that is dysfunctional that is causing that behavior? And then we can say, well, let's treat that circuit. And that will treat the behavioral dysfunction regardless of what the genetic etiology was. And so I think this is a really exciting approach that I and others are taking to help people with rare diseases sort of benefit from collective knowledge from all these rare diseases that cause common symptoms. So I'll tell you today about the work that I did as a postdoctoral fellow that's really the foundation for the proposed studies. I used mouse models to to sample different etiologies for neurodevelopmental disorders. So I used a drug exposure model, so in utero exposure to valproic acid. It's a commonly used anti-seizure medicine and mood stabilizer. We studied the Fragile X knockout mice, and then a rarer form of autism due to differences in the CNTN-AP2 gene, which is a cell adhesion molecule. So we're really sort of sampling from across the gamut of types of genetic disorders. And what we identified is that when we zoom in to particular circuits in the prefrontal cortex, one of these parts of the brain that's super important for, cognition, executive function, social interactions, we found that there was one circuit that was dysfunctional and the other circuit really wasn't. And then we asked, well, how does this circuit function? That might be okay. That might be interesting for, cells that are being recorded sort of in an artificial environment. But what about in the mouse? And so we recorded the activity of these neurons in mice and found that the circuit was dysfunctional. And then we used this approach called optogenetics, where we used neuromodulation, we modulated the activity of those neurons. And we found that it influenced how the animals engaged with each other socially. And so taking this approach from going from multiple genetic etiologies, identifying a common circuit phenotype, and then probing the role of that particular circuit in the behaviors that we want to be able to treat. So this proved to be a successful approach for when asking about prefrontal circuits in neurodevelopmental disorders. And so my plan for the proposed studies is to ask a simple question. If we perform these same studies in the SETBP1 mice, will we find prefrontal corticothalamic hypoexcitability? Will we find that same circuit difference? And if we adjust the activity of those neurons, will that adjust the behavior of the mice? Will that have an impact on their cognition and social interactions? So this is all published work, and so I'll go through it relatively quickly and just give you sort of an amuse-bouche of each component. So first, we recorded from neurons. We made brain slices from mouse, and we compared the activity of individual neurons in those brain slices when we compared the mouse model of autism versus the control. And so here I'm showing for the valproic acid model, the valproic acid treated mice versus saline controls. And when I'm showing you here this, it's a coronal slice of the mouse brain, and we're zooming in here on these gray cells that are the pyramidal neurons that project to different regions of the brain. And what we see is when we look at all these little upticks in this signal, this is a current clamp recording, and all of these little upticks are action potentials. We see that the valproic acid mice are firing fewer action potentials. They're less active. And so I talked earlier about this idea of circuits and how it's not just where in the brain it is, but it's who it's connecting to. And so when we look more closely at this particular population of neurons, we see that it's not a single population of neurons, but rather it's made of two different categories. So these categories are broadly described as extratelencephalic or intratelencephalic. So cells that project within the telencephalon, so to other cortical regions, or to subcortical structures such as the thalamus and pons. These cells differ in their projection targets, but they also have other differences in their genetic markers and electrophysiological activity and structure. And so what we found is that it was these extratelencephalic, these corticothalamic cells that were the ones that were driving that difference, that were responsible for the differences we saw between the autism models, the neurodevelopment models, and the controls. And so I'll show you those data. So here I'm plotting the frequency of action potentials as a function of how much current we're giving the cells. So it's basically an input-output curve. And what we see in black are the controls, and then the colors are the different autism models. And we see across the board that these corticothalamic cells in the prefrontal cortex have decreased excitability. So we did not see that same change in the other population of cells. And so then we said, okay, well, these cells are hypoexcitable in these different models. How are they behaving, in the animal? What information are they encoding? And might that information encoding be different in the models of the neurodevelopmental disorders? So we took advantage of the fact that these corticothalamic cells that are dysfunctional express the dopamine type 2 receptor. So from here on out, I'll be talking about these DRD2 positive cells, which is really, for the purpose of this talk, synonymous with corticothalamic cells. And so first we asked, how do these prefrontal, I wrote thalamocortical, pardon me, I mean corticothalamic. How do these prefrontal corticothalamic neurons participate in behavior? So we are able to use calcium imaging. So we can use a fluorescent signal to tell us how active these neurons are over time, in real time, while an animal is interacting with another animal. So this is called calcium fibrophotometry, and we use GCAMP6F for the aficionados in the room, and we used DRD2 Cree mice with a Cree-dependent promoter in the adeno-associated virus that we injected into the prefrontal cortex. So what we found is that in the autism model mice, and this is specifically the valproic acid model that I'm showing you here, when we plot the fluorescence, the change in fluorescence, which is a proxy for neuronal activity, as a function of time, and we aligned everything to where 0 is the time from that first sniff that the mice take of each other when they get together. When we plot the activity as a function of time, we see that these corticothalamic cells in the prefrontal cortex activate at the time these mice meet each other for the first time. And we see this peak and then this plateau that lasts a couple of minutes while the animals are getting to know each other. However, in the autism model, in the valproic acid exposed mice, we do see that nice peak at the very beginning, but then the signal simply decays and does not have that persistent activity. And so we tested if this was due to the mice simply not interacting with during that period, and that wasn't the case. They actually continued to interact with the mice, with the other mouse throughout the time of the assay. And so this is leading us to think that perhaps the persistent activity that takes place in the prefrontal cortex that we know is important for prefrontal functioning may be not functioning in these mice. And that may have something to do with that hypoexcitability that we observed. So we've identified the fact that these cells are dysfunctional at a single cell level. When we look at how they encode activity in vivo, we find that they are not activating appropriately the way they do in the control mice. And so next we wanted to ask, well, is that important? So does the activity of these neurons actually play a functional role in social behavior? Or is this just something that happens to be different, but doesn't actually have anything to do with social behavior? And so to ask this question, we adjusted the activity. We used neuromodulation to adjust the activity of these corticothalamic cells using a technique called optogenetics. And by basically activating or inactivating these corticothalamic cells during the animal's interactions, we're able to really precisely ask the question, what is the causal role of these prefrontal corticothalamic cells in behavior. So optogenetics is sort of the really has been revolutionized our ability to probe circuits. The basic idea is that we use light sensitive proteins that that let ions across the membrane. So instead of needing to have a neurotransmitter like a chemical go from one cell to the other to open a channel, we can shine light and have that happen at our will when we shine light. And so we implant these mice with fiber optics and shine light through the fiber optic to activate these populations of neurons. So it gives us really precise control in time and in space over the activity of populations of neurons. And this really allows us to probe these specific circuits and ask what role they play in behavior. Okay, so this is my last graph. So on the y-axis, I'm showing you the number of seconds that the animals engage in social behavior. So this is just the amount of time that they spend sniffing. And on the left graph, I'm going to show you the case where we inhibited the activity of these cells. On the right, I'm going to show you the case where we activated these cells at 10 cycles per second. So first, I'll show you the control mice. When we inhibit these cells in control mice, they didn't really care. There was no difference in the amount of time that they spent engaged in social activity. However, when we excited these cells at 10 hertz, we saw a dramatic decrease in the amount of time that that the mice were exploring each other. And the data are all in the paper, for if you want to look at details with the individual mice plotted. So we saw the same thing in the valproic acid exposed mice. When you activate these cells at 10 hertz, we see a dramatic decrease in the amount of time that these animals are engaged in social behavior. Surprisingly, when we inhibit these cells, we saw an increase in social behavior. So this was very counterintuitive because we had anticipated that activating these hypoactive cells would have been beneficial. We thought they're not as active as they should be. Let's activate them, and that should help, right? But that's not the case. Activating them further really disrupted things, and inhibiting them further really helped things in the autism model mice. And so this is a good lesson for those of us who are studying these circuits and studying, observing changes in circuits. What you find may be the cause of the behavioral difference that you're seeing, or a change in physiology might be some compensatory mechanism that the brain is engaging in to improve overall function in the face of that, whatever that primary cause is. So the fact that inhibiting these already hypoexcitable cells makes me think that these changes that we're seeing in the prefrontal cortex are in fact compensatory, and thus if we enhance them, that might be therapeutic. So what I've shown you is that these three divergent autism etiologies converge on a prefrontal circuit that is the prefrontal corticothalamic projection neurons, and that optogenetic modulation of that circuit modulates social exploration in autism model mice. And I'm saying autism model mice, but really these are models of neurodevelopmental disorders in general. So the proposed work we will take the SETBP1 mice and ask if these specific neurons, these prefrontal corticothalamic cells, are hypoexcitable. How are they activating during behavior? And how does manipulating the behavior of these neurons influence that behavior? So Thank you to the $1 Million Bike Ride. And I will leave it there and open it up for discussion. Thank you so much.